All right. So everyone knows about COVID-19, so coronavirus infection 2019. And as we know, it's a pandemic. It's caused by a novel coronavirus. Dr. Buchmeier is actually an expert on coronaviruses, so we're very lucky to have him here today. And as we all know, it causes mainly a respiratory infection, a viral pneumonia, but many cases are mild or asymptomatic, but it spreads from person to person. And an important feature is there is no pre-existing immunity in the human population, so everybody is susceptible. And we don't have a validated treatment. There's some treatment options we're going to talk about, and there's no vaccine. So everyone is really at risk. And as we all know, it's had a wide impact on our lives. Now, just for perspective, we're going to talk about the 1918 influenza pandemic. About 100 years ago, we estimate that a third of the world's population was infected, so 500 million people, about 50 million deaths. In the US, it was about 2/3 of a million deaths, about 2.5% case fatality rate. We'll talk about case fatality rate in the context of COVID. It spread around the world in 5 months, and an unusual feature was that healthy young adults were getting sick and dying of flu. Now, important thing, if we look at the number of cases, the graph at the bottom, you'll see that the first wave started in the summer of 1918, but about 2/3 of the deaths occurred in the second wave, which was in the fall of 1918, and then there was a third wave the following spring. And so we don't know where we are with the COVID pandemic, but we have to keep this in mind that the first wave may not actually be the worst wave. There may be a second wave later on, but we don't know yet how that's going to turn out. So what makes a pandemic strain? Well, one, the ability to infect humans. We think this is an animal, the COVID virus is an animal virus, probably from bats. Dr. Buchmeier will tell us much more about that. But it's gained the ability to infect humans. And then there is rapid and efficient human-to-human transmission. And the third aspect is we need a vulnerable human population. There's no pre-existing immunity. So we have all of these things, the perfect storm for COVID-19. We all know it started in China. It started in a city called Wuhan. I had not heard of Wuhan before this outbreak, but it has 11 million people, so it's a big city. There were a cluster of pneumonia cases right at the end of last year. And then by January 23rd, there was a citywide lockdown. But at that point, 7 million of the 11 million people in that city had left already. So you can see the containment measures were late. And we've all heard about how there's suspicion that the spread of the virus was through a seafood market. This is a live market with live seafood. There are also other live animals sold in live markets in China. And the concern is that the virus then jumped from other animals to humans. We don't know which animal actually was the last host before infecting humans. But this might have been a rare spread from one animal to a human, but then after that, it's been human-to-human transmission. Now, even though we focused on the seafood market, the thing to know about Wuhan is it's a major transportation hub in China. They have high-speed rail. This is the train station nearby, and they also have a lot of air routes out of Wuhan. So it was a focal point from which the virus could spread. So it spread rapidly around the world. The first international case was in January. This was someone who traveled from Wuhan to Thailand. And then by March 1st, we had outbreaks in Italy, Iran, and South Korea. And then WHO declared a pandemic in mid-March. And by March 26th, we had more cases in the US than any other country. So we are currently the focus of the pandemic in the world. Signs and symptoms are mainly of a typical respiratory infection, fever in most cases. This study from China has fever in 99%, but in different studies, the fever is in 80 to 90% of patients. But fever seems to be the predominant symptom, predominant sign in patients. Other things would be fatigue, a dry cough meaning that people are not producing sputum, and then dyspnea, which is difficulty breathing, and that's in the more severe cases. So this is, in a way, very similar to flu, similar to many other respiratory infections that are typical this time of the year. There have been other symptoms and signs that have been described, some gastrointestinal complaints such as nausea and diarrhea. In some patients, these have been early symptoms. Very intriguingly, they've talked about patients losing their sense of smell and taste or disorders of the smell and taste. Some reports of cardiovascular complications, and then something called a cytokine storm, which is an intense inflammatory response. These patients are very sick, and most of the deaths are in patients with this intense inflammatory response. So to give you a sense of the timeline of the infection, we think the incubation period is between 4 to 5 days. Typically, what that means is someone is exposed, and then they develop symptoms 4 to 5 days later. But it can be up to 14 days, which is why when you've heard of talk about how long someone needs to be quarantined, they usually use a 14-day period. So that's where that comes from. Patients have respiratory symptoms about after a week in severe cases. As we'll talk about, there's a spectrum of cases. A lot of cases are mild. But when patients have severe cases, they have breathing difficulty, they have shortness of breath. And this will occur about 7 days after the symptom onset. So the start of the second week, there's shortness of breath. ARDS stands for acute respiratory distress syndrome. This is where people have damage to the lungs. This occurs about the same time. This is when these severe patients need to be put on a ventilator. A hospitalization in the patients who are sick enough occurs also about 7 days after symptom onset, and typically patients stay in the hospital for about 10 days. Mild cases, patients don't need to be hospitalized. They recover after about 2 weeks. Severe cases can last for 3 to 6 weeks. Death occurs about 18 days after symptom onset. Now, this is important because when we're looking at the number of cases, you see the number of cases going up in the world, in the country every day, and you also see the reports of deaths, but the deaths are a lagging indicator. So you may actually see an outbreak contained in the country, the number of cases starts to level off, but the deaths still climb because there's this lag between onset of infection and 18 days after symptom onset when most of the deaths occur. A lot of this data is coming from China, from the outbreak in Wuhan and the rest of the country. So if we look at the spectrum of disease, it's estimated that 80% of cases are mild or have no pneumonia. So that's comforting. The problem is we don't know who's going to get sick. We have some indications, we'll talk about risk factors, but everybody is at risk for infection, including young kids. But the question is who gets the severe cases. So we think 14% are severe. They tend to have the breathing problems, the shortness of breath. Hypoxia means low oxygen in blood. And they have lung involvement on chest imaging. I'll show you a picture of some CT scans. And then 5% of the cases are critical. These are the patients who have the risk of dying. They have respiratory failure. They are on a ventilator. They have shock and involvement of many organs in their body. So here's a CT scan of a patient showing at day 4 and day 12. And the normal lung should be black. And you can already see here, this is the front of the body. This is the back. You can see some markings in the lung. We call these ground glass opacities. They tend to be in the peripheral distribution, meaning that on the edges of the lungs. And you can see, you don't have to be a radiologist to see that something has changed by day 12 and has much more involvement. So if we look at who gets hospitalized, it's well known that older age seems to be the risk. And you can see that young kids, they don't get hospitalized. Now, again, to reiterate, everybody is at risk of infection. We think the infection rate in young kids is the same as in older people. But when you get infected, your risk of having complications and severe disease is higher with age. And it really seems to be with each passing decade. The highest risk is in individuals who are very elderly. So we have in China, almost 50,000 lab-confirmed cases. In patients over 80, the hospitalization rate was almost 20%. If we look at the fatality rate, that also increases with age. So the way we measure this is something called the case fatality ratio or case fatality rate. I'll explain where we get that. But you can also see that under 50, it seems like the death rate is low. And then over 50, so all our faculty or most of our faculty, the risk goes up. And by the time you hit emeritus status, you can see the risk is very high. So that's very concerning. That's why during this outbreak, we've really tried to protect the elderly by limiting the exposure. So how do we determine the case fatality rate? So what that is a simple ratio of the deaths to the known cases. So remember, we talked about how we have severe cases, but many more cases which are less severe or even mild or asymptomatic. And so we take the known cases, and that's the denominator. So the case fatality rate is the deaths divided by the known cases. Now, the problem, of course, is that that doesn't account for the ones we don't know about. And especially in this time in this country where we don't do widespread testing, we have no idea at the bottom of the pyramid how many of these cases are there. We just don't know about them. So what we really want to know, or what you want to know in terms of what is my risk of getting sick, is the infection fatality rate, where the deaths are divided by the total number of cases. We don't know this number. We probably won't know this for quite a while. After the outbreak, they'll do serologic testing. They'll look for antibodies to see who's been exposed, and they'll have a better idea of the total number of cases. But the case fatality rate for now would be the known cases, and we take the deaths divided by the known cases. So there is some useful information, particularly in situations where we have much more information. So an example of this, you've heard about these cruise ships where people were stuck on board and there were lots of infections. So the famous one is called the Diamond Princess cruise ship. There were 3,711 passengers and crew, so basically a floating city. And we now know that a passenger boarded the ship in January 2020 with symptoms that started the day before. And then what happened is they quarantined the ship, and they tested all the passengers. So this is an unusual situation where they did testing on everybody. So we now know there were 712 confirmed cases. So you can see approximately 20% of the people on the ship were infected. And at the time of testing, 318 were symptomatic. So about half of the confirmed cases are symptomatic. And of these, 10% went to the ICU, and there were 9 deaths. And then, so we can then calculate the case fatality rate would be the 9 over this 381, right? But we know that at the time of testing, 331 were asymptomatic. They didn't have symptoms. They were only detected because a test was performed and was positive. And Dr. Buchmeier is going to talk about testing for COVID in his portion of the talk. Now, everyone is very interested in these asymptomatic cases. One, what proportion of infections are asymptomatic or mild? In a way, that's encouraging. But the issue then is, are those people spreading infection? And I'll have more to say about that at the end. But we now know that when people are asymptomatic, most of them are actually pre-symptomatic, meaning that they haven't shown symptoms yet, but eventually they do. And this is a 1 to 3 day period before they have symptoms. So we now know that of these 331 who are asymptomatic, they then followed them after the testing to see how they did. And it's estimated that the actual number of true asymptomatics was 18%, meaning that at the end of the day, 18%, so call it about 20% of the individuals didn't have symptoms and 80% did. And you'll see numbers in the media, they're talking recently about asymptomatic cases of 25 to 50%. The answer is we don't really know, but this is as good data as you can get right now, that about maybe 20% of the people are asymptomatic, but most of those people are pre-symptomatic. They haven't shown symptoms yet. And then there's at the bottom of the pyramid, these people who remain asymptomatic. So the infection fatality rate would be the 9 then over the 712. But in most cases, we don't know the 712, the total number of cases. So then if we look at the case fatality rate for COVID-19 as far as we know, and again, most of this data is coming from China, but it turns out the case fatality rate varies from country to country. So the estimated case fatality rate is 1.4%, but as you can see, again, it's much higher in those who are older. In this case, the cutoff was 60 years old. And then the estimated infection fatality rate was a smaller number. Again, they're estimating the total number of cases here. So in Italy and Spain, the outbreak has had a very high case fatality rate in the 10% range. There are a number of reasons that have been speculated. One is that they think that the population in Italy that was affected were mainly older individuals. You can see how that would skew it a certain direction. The number we think we're seeing in the US is 2.2%. Also, we've seen in many countries the case fatality rate was high in the beginning and decreased later on as people gain experience in terms of dealing with this. So how does this compare to other infections? Well, the standard that we look at is seasonal flu. This is flu that comes every winter. And COVID-19 has a much higher case fatality rate than seasonal flu. You can see all ages, 2.3% for COVID-19 versus 0.1% for seasonal flu for the last full year, 0.1%. So in the order of 2 to 2.5 times higher for COVID-19. And the same holds for different age groups. Seasonal flu also has a higher fatality rate for older people. You can see that the rate is much higher for COVID-19. If we compare this to the 1918 influenza pandemic, the numbers that are cited are a case fatality rate of over 2.5. So we're probably in the same range, COVID-19, as the influenza 1918 pandemic flu strain. If we compare it to other outbreak infections we've had recently, like SARS, MERS-CoV is another coronavirus called the Middle East Respiratory Syndrome, and Ebola, these have very, very high case fatality rates. Okay? So you could say that COVID-19, higher case fatality rate than regular flu, but it could be worse. The case fatality rate also depends on the host. So you may have heard that there are high-risk individuals, meaning that not just age, but also certain pre-existing conditions increase your risk of death. And these conditions include cardiovascular disease, diabetes shows up in a lot of studies, chronic respiratory disease, hypertension, and cancer. So these are not surprising. Most of these are actually risk factors for death from influenza as well. Now, a slightly comforting thing is, if we look in Italy, patients who died had an average of 2.7 pre-existing conditions, meaning that a lot of people have, say, hypertension, and they're wondering, oh my goodness, am I at great risk? And the answer is that you're at slightly higher risk, but it's really the people who have a number of these pre-existing conditions. They are at very high risk. So I'm going to look at some maps and graphs and tell you about some websites that are very useful. Johns Hopkins has a COVID-19 interactive map. It's updated. This was from last night. The numbers are already higher today. So today, we're probably going to break the 1 million mark in terms of worldwide cases. And the number of deaths today is probably going to go over 50,000. But again, this is real time. This is changing all the time. They have a list of recoveries, about 200,000. And they also show hotspots. You can click on the hotspots and get detailed information. So we can see here that China was the original hotspot. We now know that Europe had a lot of cases. Some of them are still raging, especially in the big cities. And now we've become the epicenter of the outbreak. Not so many cases in the Southern Hemisphere yet. There are some cases in Australia, not much in Africa. We're worried about Africa because of limited medical resources there. Here, another way of looking at different countries. This is the number of new cases. So this is each day, how many new cases are there. This is total number of cases, not adjusting for population. You can see we have many more cases every day than other countries. This is a site called Our World in Data. It's a very good site. They have data not just for COVID-19, but other things as well. I like this graph because they do a 3-day average. So it evens things out. But you can see that the number of new cases in the US, over 20,000 a day, is surpassing all other countries. So we are the epicenter. Now, partly we're a big country, but partly the outbreak is not contained here. You can see countries like China and South Korea, they're not reporting many new cases. One of the questions is, are they being totally forthright about the number of new cases, but they appear to have contained their outbreak. If we look at the map, this map is already slightly out of date, but the darker it is, the more deaths there are. So again, this is now a lagging indicator. And China initially had most of the deaths, but the hotspots really for deaths have been Iran, Italy, and Spain. And we're now seeing lots of cases in the Netherlands, in the UK. And this map is outdated because in the US now, we're a darker shade because we're having more deaths. And that will increase over time because, again, there's that lag between onset of infection and deaths. If we look at deaths another way, this is by country, but the interesting thing about this graph is, one, you compare different countries, and so you can see deaths for China have leveled off. And this seems to be the case for every country with an outbreak. So we talk about Iran and Italy and Spain as initial hotspots, they're all leveling off. Here we are in the US, we're still shooting up. What is nice about this is you can see the slope of the graph tells you how rapidly the number of deaths is doubling. So if it doubles every day, it's here. Every 2 days, it's here. Here's every 3 days. So in the US, we're somewhere doubling every 4 to 5 days. I keep looking at this every day to see if this is leveling off. It's not leveling off yet. So we really haven't contained things. And there are some countries like the UK that also have this rapid slope still. If we look at hotspots by regional information, because sometimes with countries, especially countries as big as ours, it doesn't give enough information. So Lombardia is in northern Italy. So again, if we refer to China, the number of new deaths has gone way down. In Italy, it's leveling off. Spain is leveling off. But here are the new outbreak places. New York State, we've heard about. Certainly Louisiana around New Orleans, we think that was because of Mardi Gras. Florida, especially South Florida, we know there are lots of cases. Belatedly, they issued a stay-at-home order yesterday, very late in the game for Florida. Michigan and Detroit. So there's certain hotspots in the country that have a lot of new cases and new deaths. I like this graph a lot. It's called an epi curve. It's graphing the number of cases by the date of onset. So this is giving us an idea each day how many new cases are there that we know about. And the problem is the more recent data, we don't have all the reports. So in this area here, you've got to take it with a large grain of salt. But what I'm looking for every day is, have we peaked and is the peak going down? And the bottom line is we haven't peaked yet, and there tends to be some lag in reporting. So it will look hopeful, and then the next day they'll update the numbers, and I'm not seeing the peak that we're looking for. But what we're looking for here is for the cases that are reported, at what point will we actually peak and the graph will go down? Now, if we look at states in the US, the way this is graphed is it's graphed according to days after the 10th death. So the outbreak hasn't started at the same time in every state, but they've set the start point on the graph as not a chronological time, but time of the outbreak. So Washington has had the outbreak for the longest in the United States, and you can see that their daily growth rate of deaths is now leveled off to about doubling every week. And we're doing a fairly good job in California. We've also had the outbreak for a long time. You can see that the slope is much higher for New York. Okay? But again, you see the hotspots, Michigan, Louisiana, Florida, now Georgia. So the story in the US is that we're going to have multiple epicenters, and some countries will be hit harder than others. One of the issues is in terms of the countries, the states that are being hit, how much is due to their social distancing measures. But another feature may also be big cities. So it appears that big, dense cities are at great risk. Now, we have a huge city to the north of us, LA, but in many respects, it's more spread out than some of the East Coast cities. But there is risk, it seems to be, in large cities. I was joking that some of the things that people make fun of Southern California about, the fact that things are so spread out, the fact that we drive everywhere, you never see people walking in the street, people live in these suburban homes. At times like this, these may actually be protective. So locally, if we look at California, the darker would be the more cases, and actually the more cases. And you can see the hotspots were Northern California. If anything, it was helpful for us that we knew about the cases in Northern California and Seattle, and that got us going early. So in retrospect, you look at the governor's stay-at-home order, California was very early in the game in calling for that. So cases in San Francisco, especially the South Bay, and then also cases in LA. LA has the most deaths in California by far, 66 deaths. And here we are in Orange County. I'll give you the Orange County numbers next. Another way to look at cases locally, we see these circles. The larger the circle, the greater number of cases. So again, concentrate in LA. We have some cases in Orange County. No one epicenter. It's spread throughout the county. So here are our numbers. Orange County Healthcare Agency, which is the name of our public health department, has a very good website for this. They give numbers every day. If you're like me, you start becoming addicted and checking that thing multiple times a day, even though they update it only once a day. This is the new cases every day. So yesterday they reported over 100 new cases. You can see we're still on an upward slope. I was very optimistic when I saw these numbers here a few days ago. I thought, okay, the peak's over. Some of this is artifact over the weekend. There's less reporting, maybe less testing, but you can see that we still have more cases every day. But the total number of cases that we're seeing is relatively small in Orange County. We have 600 cases as of yesterday. Compared to other places, this number is relatively small. If you look at cities in Orange County, Irvine has 57, but it really doesn't stand out from other cities. If anything, these tend to be the largest cities in Orange County. If we look at distribution by age and sex, it pretty much follows what we see in the rest of the country. An important point is that if we look at age, you can see that people under 45, so blue, purple, and green, and pink, account for about half of the cases. So again, the important message here is that just because you're young doesn't mean that you can't have cases, you can't have symptoms, okay? Again, the greatest risk of death and complications is in older individuals, but young people at risk. And we've heard of cases now, outbreaks where people went from spring break to Mexico or to South Florida and basically gathered and infected each other. Young people are not immune. We don't know why, but there seem to be more cases in males than females, a slightly greater number in males. Again, we don't know the reason for that. So let's talk a little bit about how this is transmitted. I think people know a lot about this already, but an important point is this is human to human. There are no animals involved in the transmission right now. Okay, so the virus has crossed over to humans. And it's mainly to close contact. So what do we mean by that? It's basically the people in your household, the people that you do things with. And the root, because of this is a respiratory infection, respiratory droplets, the viruses are in the droplets. Now, there's an important distinction between respiratory droplets and what we call airborne droplets. The difference is the size of the droplets. Respiratory droplets are relatively heavy, and so they don't stay in the air for that long. And so for COVID-19, for influenza, we believe that most of the transmission is through these respiratory droplets. And that's where you've seen the talk about staying 6 feet away. We used to say 3 feet or 3 to 6 feet, but now it's become 6 feet. So this is a very interesting picture showing someone coughing, and you can see these droplets. It's within this period, this distance, this 6 feet. So that's where the greatest risk is, and that greatest risk would be face-to-face contact within that time. And so what happens is someone coughs or sneezes or talks, and they spew out these respiratory droplets. They have the COVID-19 virus. And then if those droplets land on someone else's mouth, nose, or eyes, then they can inhale them and they can get the infection. Now, there's been a lot of talk about contaminated objects. And this is an indirect spread where someone with the virus may cough, or they've coughed on their hands maybe, and they touch an object, and then a second person comes and touches that object, and can they get sick? And we're all concerned about that. So now you not only stay 6 feet away from people, you don't want to touch anything in public. So the idea here is that these objects are contaminated, a second person touches it, and then they touch their hand, their mouth or nose or eyes, and they spread it. So the thing to be reassured about is this is a much less common way of spread. Okay, it can happen, but the main spread is respiratory droplets. And so you see so much fear of touching objects in the environment. The main important thing is hand hygiene. So if you touch things that other people have touched and may be contaminated, that's where you want to clean your hands. And the most important thing is you don't touch your mouth or nose or eyes with your hands. Now that's hard to do. During the course of the day, we are all touching our hands, nose, or mouth. But just to know that the indirect spread by contaminated objects occurs, but is much less efficient, much less common than the respiratory droplets. So this is, again, a respiratory infection. Now, there's been a lot of concern about airborne droplets. Another term you will see is aerosolization. So what we talk about here is small droplets that are floating around the air and can stay in the air for a number of hours. And there are a few infections where the spread is that way, and those infections are very transmissible. So to look at that, we use a term called the basic reproduction number, or R0. And what we're talking about here is from a single individual, how many other individuals do they infect? So you need at least one to maintain the infection. But true airborne diseases like chickenpox or measles have a high R0 of 12 to 18. And the R0 for COVID-19 is 2.6. So it's the numbers here do not match an airborne disease. Now, there are some instances where the virus can be aerosolized, and one way is in the hospital. There's certain procedures we do, like if we intubate somebody or do a bronchoscopy, that can generate aerosols. We've talked about aerosol generating procedures. Those you're not going to encounter in normal life. Okay, so the doctors doing these procedures have to be careful, and they'll wear an N95 mask. But normal airborne droplets, that doesn't seem the case for COVID-19. You'll also hear about studies where people have said they were able to generate aerosols of COVID-19, but they're using experimental means. So a normal person coughing and sneezing, as far as we know, there's good data, they're not generating airborne droplets. So one should be reassured that that's not going to be a mode of spread. Now, there's a lot of talk about asymptomatic individuals. So the concern here is the person behaves normally, doesn't seem to be sick, but can they spread it? So remember we talked about the Diamond Princess cruise ship and how 18% of the people were true asymptomatics, and a larger number were pre-symptomatic at the time of testing, but developed symptoms later. So we haven't had a lot of good data until just very recently. There's a new study that just came out from cases in Singapore. Singapore was early in the pandemic. They had some cases from China. So here the reassuring thing is that they identified 11 clusters of cases where there was spread from asymptomatic individuals, and the spread from asymptomatic individuals accounted for 6.4% of the cases in their total study, meaning that you can have spread by individuals who don't have symptoms at the time, but this is a really small number. Okay, so most of the cases are going to be from someone who's symptomatic, so someone who's coughing, sneezing, and knowing that those are the people who really should be quarantined at home. And again, the asymptomatic individuals, it tends to be in the pre-symptomatic period. So it's 1 to 3 days before symptom onset. And this is helpful because eventually when we do contact tracing, when we see someone who is sick and we can test them and then find out who else they've been exposed to, well, we don't have enough testing right now to do that, but eventually we'll be doing that. What we'll know now is that we need to go back 1 to 3 days before their symptoms to see who were they exposed to then, because those are people who risk for getting the virus. So there have been some studies looking at the infectiousness of COVID-19. So again, to reiterate, it seems that people are infectious about 2.5 days before symptom onset. The virus is mainly in the upper airways initially, so the nose and the mouth. And early on, even before people have symptoms, there's a high viral load. And the highest viral load is about a day before symptom onset. So that's why these pre-symptomatic people can be spreading the virus. And then the infectiousness declines rapidly at 7 days after symptom onset. And there's also concern about viral carriage. So someone recovers, their symptoms clear. The question is, do they still have the virus? Are they still shedding the virus? Based on a small number of patients, we know there is continued viral carriage, but most times they're testing this by RT-PCR. Dr. Buchmeier is going to tell you this is an RNA virus, so they can use RT-PCR to test this, and they can detect the viral RNA, but that doesn't equate to viable virus. So we don't really know the role of long-term viral shedding in terms of spread, but it would appear to be, from what we know about the viral load, that really it's the viral load early on in infection that's involved in most of the spread. So just a few questions that most of you will have about infection control, and this is going to be mostly about outside the hospital, not inside the hospital, because most of the people in the audience are not clinicians. So a question people have is, should I wear a face mask? And this is controversial, and you've heard they've even now talked about maybe everybody wearing a face mask. So here's the important thing to remember, which is that it can prevent the spread, but most of the spread we're preventing would be from the person wearing the mask. So I think most people are wearing a mask to not get COVID-19, but really where it's most effective would be if you have COVID-19, it could, if you wear a mask, it could prevent your exposure to someone else. This is coming up, say, in the hospital, where we have hospital workers who maybe have someone at home who is sick, and they're still working. So if they wear a mask, the assumption is maybe they are infected, and it's to prevent spread to someone else. Now, it's said that face masks may help you from touching your face. That's true to a certain extent because it makes you aware of your face. The face masks are uncomfortable, so they've also seen that people who wear face masks are often fiddling with the face mask, so it doesn't help. So the proper way to wear a face mask would be you cover your mouth and nose. You see people who don't cover their nose, and you only touch it when you put it on, and then you take it off, and nobody does that. So you can have a false sense of security. You're wearing a face mask, but you're still touching your face, your mask, your eyes. And so it doesn't protect you unless you use it totally properly, and most people cannot do that. An N95 mask, there's no reason to do that unless you're someone in the hospital and you're doing an aerosol generating procedure. In regular life, it doesn't seem we're going to be exposed to airborne virus. Should you wear gloves? You go to the grocery store, do takeout at a restaurant, you'll see people wearing the gloves. The important thing is the gloves don't spread the virus. In fact, if you look closely, people are wearing the gloves, they think they're protected, but they're still handling everything, touching their face and mouth with the gloves. So they're still spreading the virus. An important point, I think, about all these personal protective equipment or PPE is we have to think long-term. Okay, are we going to run out of these things before the pandemic is over? And we don't know how long this is going to go on. So yes, people need to be protecting themselves, but we don't want to run out of these things. Let's say we have a second wave this fall. What about hand sanitizers? They have to have alcohol. They're useful, especially if you're out in public. Let's say you go to an ATM machine and you touch the buttons and you know other people have touched it. So clean your hands, decontaminate them after you've done that. The likelihood of transmission from an object depends on many things. You'll see a lot of scary information saying that the virus can last for days. Basically, again, transmission from objects is not the most common route. It depends on a lot of things. One is how recently was it contaminated? How much virus was on there? What's the material? The virus lasts longer in non-porous material. But again, you can use the hand sanitizers to clean your hands after touching these things. And then, can you get COVID-19 from food? The answer is no, you cannot get it from food ingestion. So I'm going to finish up with a little bit about social distancing and a little bit about treatment. So we can learn a lot by social distancing from the 1918 flu pandemic. And I want to encourage everyone to look at this National Geographic. You can just do a Google search for National Geographic and flattening the curve. And it shows different cities and how they responded to the 1918 flu pandemic. So the colored area here is the time of social distancing measures imposed by the city. So some cities started late and had short social distancing, and those cities had high peaks. And examples of Philadelphia and San Francisco. Cities that had longer periods of social distancing and started it early, their peaks were flattened. So New York at the time had one of the lowest death rates on the Eastern Seaboard. As we know for the COVID pandemic, they have the highest death rate. St. Louis is an example where they started early, they flattened the curve, they relaxed restrictions, and then they had a second wave. Okay, so I know the stay at home, shut down the lab, these things affect our lives, but basically they're important, they're having an effect, and we can relax the restrictions too early and then have a rebound in number of cases. So we look at the measures in 1918. Remember, in 1918, they didn't have antiviral treatment. There were no vaccines against influenza. So basically, in many ways, it's similar to what we're dealing with today. So they closed schools, churches, and theaters, banned public gatherings. They staggered business hours, so not everybody was going to work at the same time. They quarantined households where there was a known infection. They isolated sick individuals. These are all things that we're doing today. I was a bit surprised to see that they banned funerals, but when you think about it, funerals are a gathering. There's lots of hugging and crying. So funerals were banned in many cities. In San Francisco, a health officer shot a man for not wearing a face mask. He shot him twice. He also shot 2 innocent bystanders. Okay, we haven't reached this point yet. Okay, very, very quickly about treatment. The bottom line of treatment is there are a few things we're trying, but nothing that we know that works with good data yet. So remdesivir is an adenosine analog. So the virus reproduces its RNA with an RNA-dependent RNA polymerase. The remdesivir is incorporated into the nascent viral RNA, and it acts as a chain terminator. It was initially developed against Ebola, but it also has activity against other RNA viruses. This is an investigational drug. We're hopeful about it, but again, we don't have a lot of data. Chloroquine, or hydroxychloroquine, is an antimalarial drug. It has good activity against many viruses in cell culture, but there's been no benefit shown for other coronaviruses in animal models. In fact, there's a non-human primate model of a different virus called the chikungunya virus, where chloroquine actually made these monkeys worse. China is reporting some clinical efficacy in clinical trials. They haven't published this data in peer-reviewed journals, so we don't know yet. It's being used, but again, we don't know how good chloroquine is. Tocilizumab is a monoclonal antibody. It's against the IL-6 receptor. It's been used to treat rheumatoid arthritis. It's an immunosuppressive, so it's been used for cytokine storm. It's being evaluated against COVID-19, even at UCI. I think the greatest hope is passive immunization. So this is taking plasma from people who've actually recovered from COVID-19. This is also being tried in many centers in the US. This is probably our best option right now. But again, early days, we don't know how much it works. And then I'm going to end with one last slide. There's a smart thermometer out there, which has turned out to be very useful. So people take their temperature. The information is reported to the company. You can also put in if you have symptoms. And they're able to spot hotspots of new fever very quickly, faster than any other way. So they were the first to identify hotspots in New Orleans after Mardi Gras and South Florida. And they reported to the CDC, and the CDC said, oh, we don't know about that yet. And then a couple of days later, it was clear there were more cases in Florida. So there are some new things going on. There's an app that's being introduced in Germany where by Bluetooth between your iPhones, they're able to tell who has contact with other people. Anyway, I'm going to pass it on now to Dr. Buchmeier, who's going to tell us about the basic science aspects of the disease and the virus. I need to stop sharing. Okay, over to you, Dr. Buchmeier. You have to unmute.
There we go. Okay, my video is on. Can you see me? I'm seeing Ming still. Okay. No.
We see your video, but I don't see your presentation yet.
No, my presentation's on the desktop. I was just trying to get my image up there. You want to leave this meeting? No, I don't want to leave this meeting. There's share screen. Okay. The presentation is right behind me on the board, on the desktop. And no, I don't want to share Rosa Andrade.
You need to share screen on the bottom.
I'm there, Rosa. I'm there. Share. I just have to, I had it on my screen already.
You got it. Great.
So let me go back. Now it's not responding to, there we go. Okay. Interesting, it's not responding to the forward and backward cursor. There we go, now it is. All right. All right, so Ming has asked me to talk about the virus, and this will be a combination of experience with this virus, which is very limited as of now, and experience that I've had with SARS, where we had quite an active group working on SARS and published a lot of papers on it. Really, we're studying it at the molecular level, so we got pretty down and dirty on it. And with mouse hepatitis virus, which some of you may know because Tom Lane's lab is also working on mouse hepatitis, but what you may not know is that Tom postdoced in my lab and learned the system there. So the virus is part of a group. The coronaviruses are certainly not a surprise to us in terms of respiratory disease because we've known about them for some time. They're a very useful laboratory model. They're a very interesting infection, a very complex but fascinating mode of replication involving some aspects of RNA replication that you don't see in other viruses except in this group. I'd like to get one thing out front, and that is that this virus is referred to variably as 2019 nCoV, or it's named in as COVID-19, or it's named as SARS-CoV-2. And the official name for it, if we care about officials, from the study group of taxonomic viruses is SARS-CoV-2, and that refers to the virus. And COVID-19 it really refers to the disease caused by the virus. So it's important. You'll see it really discussed both ways, and just consider the two to be talking about the same topic. The viruses themselves are representative of a larger group, which we call the nidoviruses. Nido because they have nested messenger RNAs, and the nested set in Latin apparently is nido. It's not something that you would necessarily know intuitively. But what I will point out here is that the viruses that we're talking about here, SARS, SARS-CoV-2, and MERS, are all members of the class 2B nidoviruses. So they're closely related viruses. Also in that group is mouse hepatitis, which you can see here, human coronavirus HKU1, and there's also a human coronavirus HKU2, and then other coronaviruses, including OC43, which causes common colds, and NL63 and 229E represent a different group. They represent a more distant group. So the point is that these are all closely related viruses. And when we talk about the difference between CoV-2 and SARS-CoV, we're talking about a 5% difference in the genome, total genome. So it's not a big difference, and it certainly is easier to understand what's going on if you just keep that in mind. Notice that a lot of these viruses also are called SARS, but they're labeled BT or RT, and BT would mean bat, and RT would mean rat. And there are different origins, different hosts for these different viruses. You have hedgehog coronaviruses, a murine, human, ratus, etc. So just keep your phylogenetic mind open on these, because there is a lot of material known about them at this point. How did they get into humans, or how do they typically get into humans? We have bats that are infected and show no symptoms, or if they do show symptoms, they're only mildly affected because they survive and they can fly. How do they get into humans? Well, if we can look at three examples here, we can look at SARS, we can look at COVID-19, SARS-2, or we can look at MERS. And these are all related viruses which are really hosted in the natural host of bats. So you have a virus which is in the bat and crosses into a human. And that is an important question, how that's happening. We think that this is happening in the live animal markets for SARS and for CoV-2. And the reason is because there is a strong association with the live animal markets, both in 2003 and 2019. MERS is a little different story. MERS was found in the Middle East. It's in bats. And the intermediate host there turns out to be camels. And so when you have an infection in the camel in the Middle East, that's an infection that's concentrated in a very large mammal. And the clusters of MERS appeared to sort out to areas that were associated with camel breeding facilities. And so it has been sorted out. The number of cases was relatively few, but the mortality was very high at about 35%. Look at the conservation between these two viruses, and I think it will give you a better idea of what's going on. If you look at SARS-CoV, where we have the most information, this is basically the genome, and this would be 100% more overlap with itself. If we look at SARS-CoV-2, we find the same basic construction. Genes are in the same place, and you have a number of genes that are very important. I'll come back to those in a moment. And then you have the spike gene. And the spike is the large, very prominent glycoprotein that's on the surface of the virus and carries out the activities which recognize the cellular receptor and trigger viral entry into the cell. And note that the N-terminal half of the spike is conserved. Oh shoot. Okay, and that is what we call S1. So S1 is conserved. That's the apical portion of the spike. The proximal portion, which is close to the membrane, has some more variability in it. And notably, this red patch here would be a place that indicates another proteolytic cleavage. So you have a proteolytic cleavage here with at the brown-red junction, and then you have a second proteolytic cleavage here, which actually is at the margin of the fusion peptide, and that's what activates the viral fusion in this case. And that's somewhat different from some of the other coronaviruses, which rely on a single fusion. That would allow the virus to enter via the endosomal route, because that's an acid pH triggered fusion. So enough on that for now. The nucleoprotein is highly conserved, the M protein is highly conserved, and these are two other structural proteins. And so it's important, and the E protein is a third one, which actually is highly conserved and forms an ion channel. So if we go back over here, the general organization of these viruses is that the genes on the left side of this border are the genes that are involved in modifying the host response and in constructing the necessary atmosphere, or taking over the cell and constructing the necessary environment to allow the virus to replicate. The genes on the left side, and this is a separation which involves only a frame shift at this point. On the right side of the slide here, you have a number of molecules. The reverse transcript, or the RNA transcriptase is here, RDRP, RNA-dependent RNA polymerase, the helicase, the exonuclease, and other genes, the N-endo-U. This N-endo-U is important because this is part of a mechanism that this virus has to edit its copying. That's important because this virus has a genome of 31 to 32 kilobases, and that's too large to sustain without some form of proofreading. And this is a very primitive form of proofreading. If we look down at MERS, you see that there's much more diversity between MERS and the other viruses, looking at this table on the side. So the point is here that SARS-CoV and SARS-CoV-2 are very, very similar in terms of their sequence and in terms of their function and in terms of their structural organization. From the SARS studies, we can present this kind of an association. We've worked out what the replicase complex consists of, and this was done by both nearest neighbor analysis and mud-pit and a number of other technologies, and by crystallography. And you can see that this is a very complex replicase. It includes peptides from the NSP3, NSP1, 10, 9, 7, 8 copies of this 7-8 molecule, 13, 14, 16, 12, and NSP15, 6 copies of that. So that the replicase complex is far from simple, but it involves methyltransferase, 3' 5' exonuclease, and helicase molecules, in addition to the RNA polymerase and the endonuclease. The structure of the virus looks like this. You've probably seen these in the news by now, but you have the S protein here with the S1 and the S2. You have the M protein, which is a protein really that stabilizes it. It's what we would call the matrix protein in other viruses. And you have this E protein, which is an ion channel, which penetrates the membrane. And then internally, you have the RNA protected by a nucleocapsid core. And this is wrapped around the inside of the particle, giving you what looks sometimes like a donut shape in the collapsed particles. I want to thank Alex Bowen here in Bert's group for his work on the vectors and providing the following slides. We obtained vectors from Thomas Gallagher at Loyola University in Chicago. And Tom, again, was another postdoc in my lab years ago, between 1988 and 92. And Tom is a very, very excellent biochemist and molecular biologist. And he very quickly produced a number of constructs. And what we're looking at here are SARS-CoV and SARS-CoV spike protein expression vectors expressed in HeLa cells. The virus has a broad range for a number of human cells, including HeLa cells, including monkey cells, if you want to work in vero cells, or in other cell lines, mostly human. The blue is DAPI, and the red is the C9 epitope tag. And what you see here is very characteristic fusion of adjacent cells, so-called syncytium formation, by the action of this glycoprotein on the surface of the cell. And so once you transfect a single cell, what it does is recruits in additional cells into that nucleoprotein complex, and by that means it can actually spread from cell to cell before it exits the cell. In other words, during the course of replication, it can spread and produce that much more virus because of that. That adds to a very rapid spread. The one-step growth curve on this virus is about 12 hours. So it's, even though it's a very large genome, very complex virus, it gets down into the range of polio or a smaller RNA virus. Sorry about that title, but the same thing, just some more examples. Syncytium formation at 24 hours with SARS spike and glycoprotein expression vectors. And what we see again is more syncytium. You can see those by DIC, or you can see them here by fluorescence. And the red, of course, is expression of that C9 epitope vector, which is substantial. And again, in HeLa cells, 24 hours after infection, and again, just additional examples. So SARS coronavirus, I'm going to take you back in history. SARS coronavirus was identified as a causative agent for an outbreak of atypical pneumonia originating in Guangdong, China in November of 2002. It rapidly spread to 26 countries, involving about 8,000 cases and 774 deaths. That seems rather pedestrian now, but what it did at the time was really shock the world. It had major impact on the countries that were affected, including Canada, including countries in Asia, and about 50% of the mortality in this was in healthcare workers. So the virus really had a serious impact, and we could consider that a warning shot. The virus is hosted by the horseshoe bat. It's a common bat in Asia. The palm civet was kind of an innocent victim in this. The palm civet is also susceptible to the virus, and what happens in the palm civet is that they carry the virus, and they may be an asymptomatic carrier or only mildly symptomatic. And the palm civet also is traded in these live animal markets. And so the spread to humans was through the live animal markets into humans. And so we had this chain of transmission that was set. The virus then can spread from human to human by upper respiratory tract infection. The disease features of all of the coronaviruses are pretty well known, and many of these cause mild to up to moderate upper respiratory tract illnesses, common colds, severe respiratory infections, etc. And only the SARS viruses, CoV and CoV-2, are the ones that are really associated with life-threatening upper respiratory infections. MERS coronavirus is probably associated with severe upper respiratory infections initially, but it also involves major organ involvement, including kidney failure. And so you have gastrointestinal symptoms and damage that really carry the disease to a higher level of morbidity. Transmission is by aerosol, coughing, sneezing, close personal contact, and MERS coronavirus is not completely understood, but it does involve camel to human and then human to human from there. Interestingly enough, there was a survey a few years ago that was published about camels in Egypt, and there was about a 95 to 97% seropositivity rate among the camels, but no documented examples of human transmission. So there are obviously some things we don't know yet about that spread, and because the disease is episodic, we don't often have a chance to study it. If we look at risk factors and things that are involved in transmission, certainly close person-to-person contact, age and pre-existing health conditions, and drug abuse, and smoking was a major one. There are no vaccines and no licensed antivirals available yet for any of these coronavirus diseases. The virus itself looks something like this, and this is an example of one of those that has kind of a collapsed looking center. And that's because of dehydration. Here's another one that may be an empty particle, but you see the very prominent spike on the surface. The nucleocapsid wound around inside, and then the other proteins involved here. If you look here, these are cryo-EM images. I'm getting a flashing light here. What does that mean, Rosa? Chat too. I'm not going to leave the meeting. Okay.
You had a question about the animal models for SARS and COVID-19.
Could we wait until the end, please?
Sure.
I'm already limited on time. Thank you.
Mm-hmm.
Okay. If you look at these from the side, you can see that they're very pronounced from the membrane to the apex of the spike. What's interesting is on the inside of the membrane, you see that there's an offset between a nucleocapsid unit here and the spike. We found no direct interaction between the spike and the nucleocapsid. And all of the interactions are mediated through that M protein, which sits in the bilayer here. Looking down at the spike, you can see the trimers. And these images were taken back in 2004, so they're pretty old. Here, you can see the trimers. And then if you look at the nucleoprotein lattice, it looks something like this. This is just from the Fourier transform. And then finally here, if you look at the membrane in detail, you can see these railroad tie-like structures, and those are the M proteins which protrude through the membrane. If we look at the histopathology, and this is important in terms of certainly a postmortem identification of damage, what you see is, once again, you see syncytium formation in the lungs. And you see in those syncytia, you'll see viral antigen expressed, and that is the red. And this was done by Sharif Zaki at CDC. And then finally, also in sputum, you can easily identify viral particles in sputum. This is a field here that's expanded on from over in this area. So that this is an easy virus to see. When these images were seen, the idea that this was a coronavirus was very quickly achieved. And by comparison with other viruses that were in the stocks and at CDC, they were able to identify it as such. So I think we are probably, oh no, there we go. Okay, the diagnosis of this in current treatment or current medicine is first to swab the patient's nose or throat. The viral DNA is amplified by RT-PCR, and using a variety of different test kits, you can use different primers to study whether or not you have the presence of a SARS RNA. The initial results were false negative because of either low viral load or poorly designed primers or sample handling and delays in testing. Part of the problem there was that CDC was using a coronavirus generic primer rather than a specific primer. And so they weren't really picking up a high enough homology and amplification to be able to definitively say this was a SARS virus. We looked for SARS-1 because that was the one we knew. Obviously, you look under the streetlight when you lose your keys. But there were other problems in sample handling, in the number of kits available, and other problems at CDC that slowed that response down. And that is debated consistently in the news. COVID-19 diagnosis also can involve an Abbott rapid test, and that's a point of care test. You have a nucleic acid amplification test. It yields positive results in as little as 5 minutes, and negative results in, I think that says 3 minutes. I'm sorry about this formatting. Influenza, strep, and respiratory syncytial virus can also be excluded at this point.
Negative results in 13 minutes.
I'm sorry?
Negative results in 13
minutes. Yeah, okay. That didn't show up when I was previewing this. Sorry, Bert, or sorry, Ming, that was Ming's slide. So the FDA emergency use authorization for COVID-19 exists, and I think we're starting to get our testing square now. There is a problem that's arisen, and that is that there are a lot of generic tests around that people have invented, either homebrewed or gotten from other companies or gotten them from Korea. People have been detected in going through airports with hundreds of tests from South Korea, and they may or may not be appropriate here in the States, but regardless of whether they are or not, they're not approved by FDA or by CDC. So chances are that the results from those can be used for diagnosis and for charging a patient, but the insurance company may not recognize them, and they may not be recognized as a valid result without additional testing. So there is a problem that comes up with that, and people are charging as much as $3,000 per test for that information. COVID-19 diagnosis there has also triggered the development of rapid tests, lateral flow immunoassay, for example, that detects viral antigens. And that's like the rapid influenza or strep test. And you have a nasopharyngeal swab. 15 minutes, you'll have a single or a double exposure line for a positive or a negative result. And that's what some of us kind of refer to as the pregnancy test version of the swab test. But it's probably a valid test. I don't know that it's really been studied to that extent yet. So coronaviruses then are enveloped. They're single-stranded RNA viruses of 21 to 31 kb. Mouse hepatitis virus is actually the prototypical virus, and it studies a lab model for multiple sclerosis and for kidney disease and a lot of other things. There's also porcine gastroenteritis virus, turkey coronavirus, chicken infectious bronchitis virus, and bovine diarrhea virus. And what's interesting about these, plus the feline coronavirus, is that there are vaccines for all of these. And you can debate whether or not these vaccines are 100% appropriate, but they certainly seem to work for the veterinary community. Human coronavirus 229E, HKU1, and NL63 cause minor diseases and haven't warranted vaccine production yet. And then finally, SARS, it was an epidemic that we thought we had eradicated. And so the surprise of another SARS was remarkable to some. Middle East respiratory syndrome had 827 cases and 287 deaths. So you can see that it was treated much more seriously. These are just a couple of other images from CDC and from Alex McPherson's lab by atomic force microscopy. This one would be MHV. SARS is capable of mutations in the course of an epidemic. And I think it's important to point that out because we're starting to see variation in virulence. We're starting to see variation in the case history of patients, and that could be significant in terms of mutational groups. And just to point out, there were 3 clusters of mutations that occurred during the original SARS epidemic in southern China. And these involved a group which, and this is basically a limited span of about 20 nucleotides in the spike protein. It's 20 amino acids in the spike protein. And these are the mutations and as they grouped. And there were a number of isolates of virus identified here that formed a low pathogenicity group. Virus invaded into humans here, and in that course of infection, viruses were isolated from patients and from civets, forming a high pathogenicity group. And then finally, another group that was referred to as the epidemic group. And this is the patients who were infected but did not die and did not have severe infection. So that there are mutations that are associated definitely with pathogenicity of this virus. We're beginning to see a serious effort at unraveling that in CoV-2, And that has been led by Christian Andersen down at Scripps. And he has shown us, first of all, two things. One is that the virus is a naturally occurring virus. It's not something that we've created in the lab. You'll read conspiracy theories that claim that. And the second thing is that he's showing that there are very specific mutations that we have to watch for. One of the things that has been shown, in fact, is that there are two cleavages of the S1 glycoprotein. And one of those is at the S1, S2 junction, and the second one is actually in the S2 portion. And the second cleavage is critical because that releases a fusion domain. And so you have what we call a priming cleavage at S1, S2, and then you have a second cleavage that is downstream further toward the membrane, and that identifies the cleavage that activates the fusion sequence. And so you have two things that go on that are necessary for full virulence of this virus. Obviously, studying how that works is important, and looking at inhibitors of that fusion is the target of a lot of antiviral drug development. So if we go back to phylogenetic neighbors and look at how these things sort out, again, we have the group 1 viruses here, which includes canine coronavirus, TGEV, is transmissible gastroenteritis virus of chickens, FIPV, which is feline infectious peritonitis. This is the one that your veterinarian vaccinates your cat for every 2 years. Porcine endemic diarrhea virus, NL63, a human virus, and 229E, a human virus. OC43, bovine coronavirus, and on down through MHV, HKU1, and on down through this group. Urbani is one of the SARS strains, and SARS-Z3 is another SARS strain. So all of this was identified at the end of the epidemic. The virulence of this virus is attributable to its affinity for the ACE2 protein. The ACE2 protein is one of the proteins that's involved in blood pressure regulation, and it's basically on the surface of most cells, and in the upper respiratory tract, certainly, and in the vasculature. The virus has the capability of also infecting kidney as well, and that happens in serious disease. But the mutations that control that infection are really illustrated here in this article. And then finally, bats are the natural reservoirs for coronaviruses. This was a paper that appeared in about 2004, 2005, when they actually got out and did the studies. And not only did they find that bats were there, but that there were a lot of SARS-like coronaviruses. And there was a series of papers that were published between about 2007 and 2015 or 16, identifying a SARS-like cluster of circulating bat coronavirus that shows a potential for human emergence. And that's a pretty explicit title. And it comes out of a large collaboration with Ralph Barrick's lab, really at the lead. And this is something that occurred in many, many publications. I've got 4 of them that I'll be happy to give anybody who's interested in them. But these group B coronaviruses are with us. They're in bats. You're not going to get rid of it. And so we need to find out a way to respond to it. And that is basically where we're at. So how does the virus get from host to bat, to human? And that's kind of illustrated here. This is two epidemic or emergence paradigms for coronaviruses. You have a host range mutation in a virus in a host, the bat. And in random cases, you get a secondary host reservoir adaptation. That would be the palm civet, and from the palm civet to the human. Humans consume palm civets in parts of the world, and that's how the infection spreads in the wild animal markets. That then can become an epidemic strain. And from that strain, you can have adaptation to direct human-to-human infection or to a secondary host reservoir. We've heard about things like the pangolin in Asia being a secondary host. And there may or may not be an infection in pangolins, but pangolins also have SARS-like viruses in their system. So again, it's something we want to watch, but you don't need to kill all the pangolins to do that. And then finally, you can have random changes that result in direct human infection. And the human then would become the secondary host. And then the secondary host is another reservoir, which is established. That could be the camel, or it could be the palm civet. And again, you have an epidemic strain that comes out of that. So there are a couple of options that are certainly under consideration. And I think keep your mind open and watch what happens in the news. There certainly is more than adequate coverage in the news today. So that's the end. I want to thank Ben Newman. Ben was my postdoc through all the SARS work, and Ben really is still very active. He's now a department chairman down in Texas. Thank you.
Thank you, Mike. There's a question in the audience. One of them would like to know more about the animal models for SARS and COVID-19, if any. And the second question is about the sensitivity of the viral culture for COVID-19, for diagnosis of COVID-19.
Okay, I'm trying to get back to a screen where I can see everyone. And I don't know, it's too much technology for me. I'll go ahead and answer the question. Animal models. Right now for SARS, the animal models are primarily mouse models. There are other animal models being explored, and certainly we don't use palm civets for a model because they produce epidemic disease, and that would cause a huge risk to animal caretakers. But we do use mice that have a knock-in human ACE2 receptor, and those are have been developed by Ralph Barrick's lab and by Stanley Perlman's lab in this country and in other labs in other parts of the world. And they produce a reasonable infection with pathology that is a good model for MHV. Mice that do not have the human receptor are not yet directly infectable. However, it's possible that you could evolve a virus that would infect them. And that can be done rather easily with this virus. You simply do a mixed culture of cell A and cell B, and you pass it through about 10 passages, and you end up with virus that's specific for cell A and for cell B. That is kind of creating new strains that we may not want to create right now. So I wouldn't recommend that one. There are a lot of incidental hosts that have been identified, or potential incidental hosts that have been identified only through sequencing efforts. The one I think that's the most controversial and that came up in the Seattle epidemic is the house cat. The house cat has a similarity, but not an identity with the receptor, the human ACE2 receptor. And there have been a lot of speculation of whether house cats were a serious host of the virus in places like nursing homes, where cats, wander the nursing home and jump in people's laps and would be a logical spread. There has been no evidence yet, and I've double-checked this with my friends who are closest to this. Stanley Perlman, who's on the WHO panel, Ralph Barrick, who's at the center of a lot of this research, Mark Dennison, who's a clinician in Nashville, who's worked on it extensively. And none of these people know of any example of a human infection traced to cats. What there is the potential that this virus will infect cats, but nobody has really looked at that in detail. Now, it's possible that you can find something in the Chinese literature where someone has, but that's never been vetted in the international literature. So the cat situation is right now is speculative and based only on a receptor homology. If we look at other experimental animals, if you look at the deer mouse, Peromyscus, and I think we're looking at Peromyscus leucopus here, they also have a homology, and that is something that is being explored right now by people here at UCI. And I would only let them talk about that. As far as other animal models, there are some animal models that are suggested by the presence in the animal markets, but I don't think anybody in this country has gone to the extent of trying to colonize those and work with them. It's possible that somebody is in a zoologic entity, but not seriously. I would emphasize that if we can develop these models and develop vaccines for veterinary cases, I think that it is important to keep in mind that those same strategies may work. Although we do have vaccines for West Nile disease, which work in horses and other agricultural animals, and we still haven't developed a human vaccine. So, there's more involved than just a wish.
I'm going to answer the question about testing And then Mike, we'll have you answer a question about the prospects for vaccine, how likely and time frame. So for the testing, there was a question about sensitivity of viral culture. And just to make it clear, we don't routinely do viral culture for the diagnosis. So basically, we don't do culture. The main test that's being done is the RT-PCR test to amplify the viral genetic material. The problem there is these tests have not been studied, and not a lot of good studies. The studies out there are low quality. There are some issues such as the viral load is going to be highest at just about the onset of symptoms. It may be too low early on. It certainly goes down later on. And so we don't know how good the test is. There is some data saying that a nasopharyngeal swab, so going through the nose to sample instead of into the mouth, is better. Sensitivity numbers that have been reported somewhere in the range of, say, 80%. But again, there's no good data about that. The lateral flow, the pregnancy tests that Mike was talking about, it'd be really good to have a rapid test, but we know from influenza and strep, those have even lower sensitivity. So if it's positive, good, you know you have it. If it's negative, you don't know whether the test missed it. So Mike, could you talk about vaccines, the likelihood that we can get a vaccine and when?
Yeah, am I still unmuted?
You're okay, we can hear you.
Okay, good. Yeah, I think the other thing about the testing is when you test.
Yes.
Can be critical, and you have no way really of knowing. When a patient is first seen by a physician, you don't know when the infection started, and if there's a time of sensitivity peak, then you could miss it easily. In terms of vaccines, I'm pretty optimistic about vaccines because we know that we can do that in mouse hepatitis virus, and it's not that difficult. But the problem here, of course, is developing a vaccine in a limited time frame. Had we started back in 2007 when these reports started to turn up of coronaviruses in bats in Asia, then it would have been much more reasonable, I think, to begin and do a really systematic vaccine development. What we do now is we're forced into techniques that we know will work in experimental systems like purified spike protein and things like this. So one of the things that worked in SARS experimentally was that the spike protein was purified by Novartis. And Novartis used this as a vaccine in monkeys, I believe, and it was effective. And then NIH shelved the SARS work, and Novartis no longer had a contract, and that was the end of that. So if we could revive some of these old strategies, I think that would be very useful. The other thing is that there is some suggestion from studying people that were infected in the original epidemic that there is still antibody. There's been a lot of debate about the duration of antibody in individuals, but there is starting to show up some information that suggests that antibody still exists in those animals and that antibody is active against CoV-2. So it may depend on the individual, it may depend on the circumstances, but it suggests that there is some resistance between CoV-1 and CoV-2. So that's an area that needs to be explored. The quickest would be a killed protein, and that would be analogous to hepatitis B, because that has the least risk in terms of human toxicity. The longest prospect would be some kind of live attenuated virus.
We've seen estimates of 12 to 18 months. Do you think that's realistic?
I think with the emphasis that's been put on this and certainly the political will, yeah, I think that's a possibility. We may make mistakes in that. We may have some false starts. We may have some vaccines that turn out not to be what we thought they were, or that cause problems in selected group of individuals. But we need to really push it very hard.
So the thing then is that for a second wave that may come, let's say later in the year, we won't have a vaccine yet. So that's something for people to be aware of.
That's right. We won't have a vaccine yet. That's right. We will have the beginnings of some herd immunity by then. So if that's sufficient, that may slow it down somewhat, but a second wave is quite possible.
Mike, do you think that a potential mutation of the coronavirus might have an impact on the vaccine design?
Absolutely, absolutely. If you choose a protein that has already been demonstrated to be easily mutable, and and produce live virus, then yeah, I think that you have to take that into account. How would you design something? Well, you need to find another critical factor in the virus that is susceptible. One problem with the protein is deeply embedded in the membrane. It only has about 2,000 Daltons, and it's mostly 2,000 Daltons of O-link sugar, which is accessible at the surface. So it's already masking that little bit that sticks out. The ion core may not be accessible at all. It certainly is to ions, but to an antibody, maybe not, but other aspects. Many years ago, we showed that antibody to S2 was effective at blocking fusion. S1 was effective at neutralizing virus. And so there are some possibilities there to have a multi-focus vaccine rather than a single focus.
There's another question regarding, how do you think that it is possible that we might have another typhoid Mary type of situation with people shedding virus and potentially being infectious? What are your thoughts about it, since we know that people that recover might be shedding virus up to 57 days as most recent information?
I think more correctly, they may be shedding virus RNA up to 57 days. And I think that's what they're basing that on. During the SARS epidemic, a number of patients were surveyed who were leaving the hospital after recovering from the acute phase of SARS, and were followed for at least 60 days, maybe 90 days, by their physicians, and were found to excrete viral RNA for a long period of time. And they were never associated with any further spread. So it may be that we do see RNA. Coronaviruses are not just upper respiratory viruses; they're also gastrointestinal viruses, and that's just the fact with the group. And so if we keep that in mind, I think that we just have to ask one more important question, and that is, does RNA equal infectivity? We know that in mouse hepatitis, the virus peaks in a very, very quick infection in the brain, but then the RNA remains present for at least 100 days after that when it's causing demyelinating disease. And Tom and I discovered that 20 years ago. So that wasn't necessarily associated with more spread of virus, but just with continuation of disease.
There are several questions related to the vaccine, but there's one. Someone mentioned if the bats are a reservoir, meaning that they're not symptomatic, do you think that it's worth studying their immunity?
Well, bats are the hosts of a lot of viral infections, and we already know that they have a defective innate response. So I think the answer to that is yes, it's definitely worth it. And CDC has a lab actually driven by one of Bert's postdoctoral fellows, Jonathan Towner, which is doing exactly that. So Jonathan Towner has colonized bats, and he is studying the immune responses and pathogen interaction in bats. So yeah, we do need to do it. But remember that flu is also a gastrointestinal disease. Flu goes from continent to continent in waterfowl. And it makes perfect sense. If the waterfowl were infected and the respiratory system were affected, they wouldn't make it across the oceans, across the Bering Straits. They wouldn't make it north and south along the Pacific Flyway. Now, I don't know about the ones in Mason Park, but those geese are pretty big. And so if the animal will not survive, remember that a fundamental rule in virology is that if the virus kills the host, that's the end of that viral genome. That genome will disappear from the gene pool. So fatality is not a desirable trait for a virus.
Another question is about the antibody-dependent enhancement. I'm not so sure about what they mean by this question.
Which question? I guess I can't see the question.
Yeah, it says, what about the issue of antibody-dependent enhancement? I don't know whether this person is still in the audience.
That's probably Don Forthall.
I really know.
And I think, yeah, there is some certainly some evidence from people who have studied the follow-up of SARS that young people don't get sick or get very mildly ill, and older people do. And that follows the same pattern as dengue. You know, in dengue, every school child is exposed to dengue by the time they start school in Bangkok. And it's only in infections later in life that they get really seriously ill. And that's because of antibody-mediated enhancement. And so the possibility is that infection with one coronavirus or infection once with a coronavirus is enough to trigger an antibody that could carry an enhancing feature. And Don has written a supplement to his grant to look at that. And that's something that I'm involved in peripherally as well.
Mm-hmm.
We don't know the answer, in other words, right now, but it's a possibility, certainly.
Mm-hmm. There's a question regarding testing new compounds against coronavirus, COVID-19. Do you know of anyone working in that field that perhaps, or I don't know who asked, but someone is interested in finding out who's working with that approach?
You know, there is effort being expended, and I'm getting daily requests from drug companies in all over the United States and even outside of the United States to help them with that. The problem is that these are, so far, they're largely drugs that are being repurposed, drugs that have already been approved for some other application, and now they want to go back and look at them for this virus. And that's something that is done a lot, and it does yield occasional additional licensing, but it's not drugs that are designed specifically to go after SARS. I would rather see somebody really target something like the SARS polymerase, which is and remdesivir does target viral RNA polymerases, but not specifically SARS. It's looking for similarity between polymerases. In an epidemic situation, you need something that's pretty high affinity and acts reasonably quickly to shut down an infection.
Mm-hmm.
But remember, the other thing that, and I tell the journalists this, and they never print it, but studying an infection is like studying a plane crash. You have so many things going on and such an urgency at the time it happens that things are missed, that the causes are not necessarily obvious. And it isn't until 2-3 years later, when you keep studying the results of that you begin to understand what really happened.
Mm-hmm.
And there's a lot that's going to be learned from the enormous amount of data that's been generated from this infection and follow-up experiments. I'm not pessimistic about that.
So, Mike, ACE receptors, ACE type 2 receptors, are important for entry of coronavirus in cells. Do you have any thoughts about whether people receiving ACE inhibitors should stop their hypertension medications? Is there any data that supports one way or the other?
Well, there has been a suggestion that people who are on antihypertensives should stop them if they think they've been exposed to a coronavirus infection, because you will compromise the ability to function. But so far it's not something that has been applied clinically, as far as I know.
Maybe I'll take that question. So the issue, again, is that the virus binds to the ACE2 receptor, and in some animal models, it's been shown that ACE2 inhibitors, which are commonly used to treat hypertension, high blood pressure, they can increase expression of the ACE2 receptor. So as Mike mentioned, the issue then is, should people stop taking ACE inhibitors for hypertension? And some societies have actually come out explicitly saying that patients should not, that the data is not settled, there's no human data, and so people should continue to take the ACE inhibitors to treat hypertension. We know hypertension seems to be a risk factor for cases. What we don't know is it pre-existing hypertension or uncontrolled hypertension at the time of presentation. So still take your antihypertensive medicine. There's no human data to suggest that it puts you at greater risk.
Mm-hmm. And one last question, because I think that we are already at 10:30. One person in the audience is asking, if someone has symptoms suggestive of coronavirus, particularly fever, but their test is negative, should they get isolated? And I'll add to that, should they get retested? What are your thoughts about it?
Yeah, so the question here is actually, one would be, what if you have symptoms and you haven't been tested? Because again, we're not doing widespread testing. And so the best thing to do is to isolate yourself at home and try to limit the spread to other people. Again, if you're a healthcare worker and you have to go to work, you wear a mask. But actually what we're really strongly suggesting to individuals is if they have symptoms, they should stay at home. Now, if you've been tested and the test is negative, again, we don't know the sensitivity of the test, but we do know the tests don't detect all the cases. So it's still better to isolate it, to quarantine yourself or isolate yourself at home so that you reduce any chance of spread. And then there was a question about why are there very few cases in India or some other countries? And there are a number of issues there. One is testing, although I think if there's a severe outbreak, you'll see lots of deaths. I think another is whether or not the virus has been introduced to the country or the region yet. So no place is immune. I think we're going to see like a domino, one after another, different regions, different countries will have outbreaks. And so, and then I think what I'm closely following is whether it gets reintroduced to other countries. So does China, do other countries in Asia have second waves? There is some early data suggesting there may be some immunity. Mike may want to comment about it, maybe short-lived in the order of a couple of years. But certainly in China, not everybody was infected, so they could certainly have a reintroduction in the second wave. So countries like India, I think it just is a matter of time, unless they have social distancing.
Mm-hmm. Well, thank you very much, Dr. Tan, Dr. Buchmeier, for this seminar. It was really interesting. I think that we don't have any more questions.
I thank you, Dr. Andrade. I think I'll make one more comment on behalf of Suzanne Sandmeyer. There is a database for SARS-2 work. So the survey was sent out. If you are doing or planning to do some work on SARS-2, fill out that database so that we know who's doing work and people can maybe get in touch with each other and collaborate. We'll probably have some kind of follow-up about this because this story is changing and developing. But thank you both. Thank you everyone for being here. Thank you, Dr. Buchmeier, for your excellent presentation on the virology and basic science. Thank you, Dr. Andrade, for being the chat moderator. I want to thank Anne Ward and Suzanne Sandmeyer from the Office of Research in the School of Medicine for helping to coordinate this presentation. Thank you, everybody. Oh, by the way, the presentation will be recorded and will be available on the School of Medicine YouTube